I'm Dana from the Red Group in Tel-Aviv University. I'd like to thank the organizers for this workshop and this wonderful opportunity to present our novel approach. Our novel approach for inferring drug targets using personalized perspective and using in silico knockouts through PPI networks. So drug target inference had an enormous interest in computational biology, as drug development is very expensive and the number of new targets is very large. The implications of any target is very large. So we have kind of a natural and essential way of computational biology to step in. And our inspiration for the work So the inspiration for the work was drug target inference used in the connectivity map project. And in the connectivity map project, they ranked small bioactive molecules according to their ability to reverse the gene expression signature of a disease. So they took the small molecule and they compared the gene expression that was received by introducing the molecule to the cell and compared it to the gene expression signature of the disease. So in this case, you can see that the molecule upregulated the underactivated genes and downregulated the overactivated genes. And so the molecules are ranked according to this, to their ability to reverse the gene expression. So we looked on mutations in the prospect of cancer cell. And what we assume is that the mutations start a signal via the PPI network, and this signal can be viewed through the differentially expressed genes in the patient's cell. And we hypothesize that if we would find drug targets or nodes in the PPI network that would reverse the effect of the mutation on the differentially expressed genes, that would be a very good target for this patient. And we will try to find these drug targets using in silico knockouts in the PPI network. So we start by simulating the disease state of a patient. We take a PPI network, and from the mutation, we propagate through the network. The propagation is a random walk with restart, very similar to the random walk with restart in HatNet. And we run it enough time until it saturates. So when we get to this state and we stop the propagation, we have a deterministic answer to how much signal was received in each of the nodes in the network. And we now look only at the differentially expressed genes of the patient. We simulate a drug by knocking out its target, so by deleting the corresponding node in the network and propagating from the mutations again. And again, we have a different propagation, and we receive the knockout-affected propagation by simulating this drug. And we do it over and over for every node in the network, so we have many knockout-affected propagations. And now we ask which is the best drug, how should we rank them? So the best drug would be the drug that would reverse the disease state propagation to the healthy state. A healthy state is simulated by a network with no mutations. So this network, PPI network with no mutations, where we choose random k nodes and we propagate from these random nodes and we repeat it n times. So in our case, we found that 1,000 times of repetition kind of give us a good cover of the network. And so by repeating this, what we actually get is a distribution of the propagation. So for each gene, a distribution of how much signal it would receive through the network through random activity in the network. So for each gene and each knockout, what we have is first a distribution of the gene in a healthy state. So how does it behave in a healthy state in random activity, and the signal in a disease state, and the signal in a knockout state. And what we want to check is whether the knockout helped push the signal of this gene towards the healthy state. So we actually count the number of times here that the knockout helped pushing the signal towards the healthy state. And then we average this over all the differentially expressed genes of the patient, and we get a back to healthy score for a drug target. So this back to healthy score for the drug target actually says, in average, how many times did this drug help pushing the signal back to a healthy state. And we ran this on HIPPY network. HIPPY network is an aggregation of databases of human PPI networks that gives weighted network according to confidence of interactions. And we ran this over AML with 174 patients from COSMIC. And we received for each patient a ranked drug target list. So for each patient, the drug targets that would be best suited for him. Now, as personal drug targets for a patient is very scarce and is difficult to validate, we had to aggregate our results. I will continue explaining in the meanwhile. So we had to aggregate our results from different patients to one full list to compare to known drug targets that exist. Okay, so before our small break, what we had is a ranked list of targets for each patient. And now we wanted to check that against a known list of targets, a known drug targets databases. So we had to aggregate the results from all the patients and receive one list of drug targets. And we compared it against drug targets from Drug Bank and against causal genes from COSMIC. And so we see here our results, our predicted drug targets, lined up from the best scoring one to the least scoring one, only in the top 100% nodes in the network. And the blue lines are hits from COSMIC, the green line are hits from drug targets. So we can see that our ranked list is very highly enriched in both drug targets and causal genes. And we wanted to assess whether the personalized approach that we took indeed help improve the results. So we built a consensus patient by taking all common, appearing in more than 5 patients, mutations, and differentially expressed genes. And we built this consensus patient, and we ran our approach on the consensus patient. And then we, again, lined up our predictions. And as you can see, the results are not significant. They're underscoring the utility of using the personalized approach. And we also tried a very naive approach of discarding the network, just taking the common mutated genes and see if they are enriched with drug targets, and they were not enriched. And that was quite a naive approach of checking that. And we wanted to assess still with some kind of personalized data. So for a mutation-specific patient, there is some kind of sensitivity experiments that were done. For the largest group of AML patients, the one that have the FLT3 internal tandem duplication, there was sensitivity experiments performed. And we took this subgroup, and we looked at the sensitivity of inhibitors in our networks, inhibitors that were experimentally validated. And we saw that the PI3K received the lowest of the 3 back to healthy score in our predictions. And it corresponded, or it recapitulated in some way, the poorly responsive to PI3K of the FLT3 ITD patients. And next in our evaluations was the MEK inhibitor, and it corresponds to what the researchers saw when they tried this experimentally, the decrease in proliferation, but the still stable line of apoptosis. And finally, the PIM inhibitor received the highest back to healthy score in our predictions, and FLT3 patients do respond very nicely to PIM inhibitor, and that led to the development of AZD1208 drug. Now, I'm not saying that the sensitivity, so this is, of course, only an example, and it needs to be validated against a large data set of sensitivity once a large data set would be available, but it is a nice trend line. And what is also interesting to notice here is the PIM inhibitor. So the PIM inhibitor does not target only one node. It targets 3 nodes, PIM1, PIM2, and PIM3. And in our approach, it's very straightforward to simulate a knockout of 3 nodes. You simply knock out the 3 and run the approach. So that's also a very nice application. And with that, I would like to thank you for listening and to thank Ortal and Eyal, my co-authors. They are here presenting in PSP, and Roded, of course. And we are all supported by the Safra Foundation.
Questions? So, yeah.
Thanks. So the score is based on the inversing the mutation pattern. So if we have other mutations that are not related to that disease, and maybe this drug can cause other mutations that are benign. So does the score change with that?
So the score is about reversing the effect on the differentially expressed genes, not the actual mutations, but only the genes that are affected by the mutations.
That are relative to that disease.
To that mutation, yeah. And so you are asking if there are mutations that are not related to the disease, would they?
If there are genes that would change their differential expression, but that's not relevant to that disease.
Yes. Okay. So great question. Would our knockout cause any peripheral effect? Would it change other nodes in the network that we did not want to change? So it can happen, yes. And it's a good question whether to try and find a knockout that would only change the differentially expressed genes and would not change the others. And that's a question of, I guess, how much do we believe to the differentially expressed cutoff? No, we take some kind of cutoff. We know that these genes change their expression, but others might have changed and just didn't make it to the cutoff. And so it's kind of a softer constraint than saying change only these expressions and don't change the others. But you could also say, let's take a harder constraint and let's choose the drug that would only affect these differentially expressed genes. And we chose not to do it because the cutoff is kind of focusing us on these genes, but is not so revealing as to other genes in the network. Yes.
The drug targets are specific AML?
The drug targets from the drug bank are specific to AML.
There are pretty large number.
Just 22, actually.
Yeah. 22 different drugs.
Drug targets, not drugs.
And how many drugs?
So I'm not sure exactly how many drugs was it. Do you know? I think most drugs had between 2 to 3 targets. So I guess about 12.
So from a computational perspective, how long does this take to run, and if you actually run it for 2 knockouts, for example?
So one propagation takes a few seconds, and we have 15,000 nodes in our network, so a few hours, something that is reasonable. But no, 2 knockouts would be, I mean, you could run it, but 2 knockouts, 3 knockouts as a way of actually exploring the entire networks becomes too long of a run. And so we only did it with the specific sensitivity where we knew the targets and were very aimed.
So a very quick philosophical question. Why do you do random walk?
Why do we do random walk for diffusions?
I mean, why do you do diffusions?
Because we presume that the mutation start a signal that would go through the PPI network. And so the mutation would affect these genes that are on the signal.
Right. I make the same assumption. And I sometimes question the validity of this assumption myself. So it's another point of discussion for the afternoon, perhaps. Not this afternoon, but probably tomorrow.
So we do have previous works on signaling that looked at the shortest path, and of course many others also looked at shortest path.
I'm not suggesting that shortest path is the answer. I'm saying, well, we are trying to model the diffusion process or the random walk process within a nucleus, maybe. But is a random walk on the PPI the right way to emulate?
No, I'm not saying either shortest path or a random walk. I'm just saying there are various methods, and it's a very good question what indeed simulates signaling.
How is this diffusion results? For example, how are the differentially expressed genes that predict, the effect on the genes? How is this dependent on possible errors that you could introduce to your PPI? Because we know they are definitely not complete. They might have some, also some false positives in there. So, did you try introducing like stepwise increasing noise in these networks and then see how this changes your results?
So it's a great question, Eva. It corresponds to what we talked about yesterday afternoon. How much is the network reliable and what can we do to see whether it indeed helps us? So we did look at what can we do without the network, and we did see a signal with patient-specific and did not see a signal with non-patient-specific approach. So we do assume that there's something there, but definitely you can perturb the network. That would be a great way of also validating this. And in this perspective, this is also why we chose the HIPPY network, because it's weighted according to how many different experiments saw the same interaction. So it's somewhat more reliable, but again, you're raising a very important point, definitely.
So,
sorry.
So you use these mutation signatures for on one of the slides. So you also have these healthy patients that you make a comparison with. What do you diffuse with the healthy patients?
We don't have healthy patients. We simulate a healthy patient. So we need to start from somewhere, right? We need a prior set. We choose k random nodes in the network, where k is the number of mutations that the patient had. And then we repeat this 1,000 times. So we just choose random starting points, and then we get the distribution.
Okay.
Yes, that's the question.
So I have probably a more philosophical question. So in this case, you look only at genes for which you have drugs, which as perturbation points, which kind of already focus your attention to where intervention may help. But in principle, if you could target any genes, assume that you say, okay, target this one, we do it. Isn't there a risk that you could then target the genes that are actually responsive and that are actually buffering the disease and kill those guys rather than the ones that are really causing the disease?
So you're asking whether we might look at the genes that are downstream of the...
I think also I'll say some immune response genes, or there are some genes that respond to the perturbation to counterbalance it. And you really don't want to bring them down. You want to bring the causative gene down, not the one which buffers the problem.
Yes, definitely. So we are worried of side effects or peripheral side effects. And this is again a question of how much you want to be specific. You want to change only these genes and not others, right? So in this approach, you could insert this very easily, so just not wanting to push them, but yeah, there's a question.
It's not exactly this. So that's part of it, that you want to change this one, not the other. But what I'm asking sometimes is, do you really want to change those one that's been changed? Because they may be big landscape of the genomic expression change, and maybe some of them are benefit to bring down to what normal was. And the second, maybe you don't want to change the expression of some other.
The philosophy is to change the entire signature towards the healthy.
Right.
And therefore, there will be no need for the ones that control the better.
Right, if you have all the signatures, then of course, but...
Okay.
Yeah, but really limited.
Donna again.
So